So animals are incredible. Some of them can fly through the air. Some of them can swim. Animals have incredibly diverse body plans. For instance, this nudibranch. Some of them can pattern their coloration in different ways, like this moth. And even what we might consider simple organisms, like the jellyfish that we see here, or a sponge. These are incredibly interesting organisms as well. And all of these animals share in common something important, which is they are composed of thousands and millions of cells, and these cells are working together to make the organism work properly. How did this all come to be? Well, that's the focus of the talk that I'm going to give you today. The work in my laboratory has to do with the origin of multicellularity. My name is Nicole King. I'm an investigator with the Howard Hughes Medical Institute and a professor at the University of California at Berkeley, and I'm excited to be here today to tell you about my research. Now, in the closing line of Darwin's Origin of Species, he remarked on endless forms most beautiful, and he was referring to the incredible diversity of body plans that we can see here. And much of his research and thinking had to do with trying to understand how do we get this diversity of organisms. And there's been a great deal of progress in this regard, largely from the work of embryologists and evolutionary biologists and geneticists working together to try to understand what are the molecular and mechanistic underpinnings of the diversification of animal body plans. But in fact, there's something else important that we need to keep in mind, and that is that animals are united by their shared ancestry. They all share a common ancestor that you can see here indicated by this red dot. And in fact, we know relatively little about the nature of that organism. We don't know much about what its biology was like or what its genome contained, and we know even less about the organisms from which it evolved. But we can make some reasonable inferences about the prehistory, the pre-Metazoan history of animals. What we can reasonably infer is that there were some important evolutionary processes that predate animal origins. And these have to do with the origin of multicellularity, the transition from a single-celled lifestyle to one with organisms that were capable of being multicellular and coordinating the activities of their different cells. So what I'd like to talk to you about today, in this first part of my talk, is what are the big questions that we want to ask when we want to think about reconstructing animal origins. And I think there are some discrete questions that we can start to address. The first is, how did genome evolution contribute to animal origins? It's clearly the case that different groups of organisms on the tree of life have different types of genes in their genomes. And what we're interested in my lab is trying to understand how changes in gene sequences and the composition of genomes might have contributed to animal origins. In addition, we're interested in understanding how genes that are required for animal development might have functioned before animals first evolved. One of the special things about animals is they have different cell types that are not found in other groups of organisms. These might include neurons or the epithelial cells that make up your skin and the lining of your gut. How did those specialized cell types first evolve? And then in a topic that we didn't expect to be studying, we find that we're becoming increasingly interested in how interactions with bacteria might have influenced animal origins. And I'm going to come back to that topic in part 2. And of course, in the background of all of this, we're interested in understanding the evolutionary implications of multicellularity. And this is a topic of research that is ongoing. Now, historically, we've been very interested, or evolutionary biologists have approached the evolution of animals and the diversification of body plans by really focusing on the fossil record. And fossils have been great. They tell us about the age of certain animal groups, and they can tell us about the shapes of some of their body parts. So for instance, these beautiful star-shaped objects are actually spicules from an ancient sponge. This is a hypothesized embryo that has recently been recovered. And here we have a fossil of a coral. And so we can see the fossil remnants of animals. But it really doesn't tell us the whole story. It doesn't tell us how animals came to be, and it doesn't tell us how cells in those ancient organisms actually interacted. to really understand animal origins, I think we need to be focusing on comparing living organisms. And so what I'm going to tell you in this first part of my iBio seminar is about an unusual group of organisms called the choanoflagellates and how they can give us special insight into animal origins. And then I'm going to tell you about how the study of choanoflagellates and comparisons with animals have started to reveal the genome composition and biology of the first animals, organisms that lived and died almost 1 billion years ago, and yet by studying living organisms, we can learn about how they function. In part 2, which I will come to later, I will tell you that some choanoflagellates can transition between being single-celled and multi-celled, and I'll tell you about how that happens, and in addition, I will tell you about how that's regulated. There are intrinsic and extrinsic influences on this process. But let me get back to this big question of how did animals first evolve, and in particular, can we focus on multicellularity? So let me remind you that animals are not the only multicellular organisms out there. We are only one of many diverse multicellular forms out there. So of course we have representative animals, but plants are a remarkable example of multicellularity. There are also green algae, the fungi, and on the far side of the slide, the slime molds, and there are probably 20 different lineages that are multicellular. And so each of these lineages has an interesting history in terms of multicellularity. And you might think that we could compare among all of these lineages and learn something about the origins of multicellularity. But it turns out that that's not possible, and that's not possible for a few reasons. One is that if we look at the cell biology of each of these different multicellular lineages, we see that their multicellularity is set up differently. So some organisms, like plants and green algae, they have stiff cell walls that mean that a cell is born where it's going to die. They're not able to move around relative to each other. Whereas animals and the slime mold don't have a cell wall, and the cells are able to move around and resculpt, and that changes their ability to form complex structures and interact with their environment. So these differences at the cell biological level also help us to understand something that we see at the level of genomes. Now, you might imagine that you could compare the genomes of different multicellular organisms and the genes they share in common, which are indicated here at the intersection, that these would be the ones involved in multicellularity. But in fact, that is not the case. The genes found at the intersection of comparing the genomes of these different multicellular lineages are the genes that are involved in basic housekeeping functions of the cell: DNA replication, translation, repair, etc. The genes that are involved in mediating interactions between cells are actually the genes that are unique within each of these genomes. Why? Why is that the case? Well, to explain why the genes for multicellularity are different in each of these lineages, I need to introduce you to a simple tree. So what I'm showing you here is a very simple tree depicting the relationships between 3 different major multicellular lineages: the animals, which are also called the metazoa, the fungi, which include the mushrooms, and the plants. And what I hope you can see, there are a few surprises in looking at this tree. First of all, it's only recently been appreciated that the closest living multicellular relatives of animals are the fungi. But the other thing I need to tell you is that by looking at diverse organisms, it has now become clear that multicellularity evolved independently in each of these lineages, and that's depicted by these yellow bars. So we think, actually, that the last common ancestor, for instance, of the animals and the fungi, was not multicellular. In fact, it was unicellular. So we have a rich history of unicellular life before the origin of these different multicellular lineages, and then these lineages evolved multicellularity independently. Well, what are we going to do? How do we operate within this framework to learn anything about the nature of the organisms from which animals first evolved? Well, the way we do that is to try to find lineages between this long extinct unicellular ancestor and the origin of multicellularity here in the animals. And we do that using a group of organisms that sits in the sweet spot on the phylogenetic tree. And these are the choanoflagellates. So choanoflagellates were discovered long ago, and I'm going to tell you quite a bit about them in the next few slides. But I want to say that the evidence for them sitting on this spot in the tree as the sister group of animals or metazoa is that they have shared cell biological features with animals that are not seen anywhere else in diversity. Phylogenetic analyses of diverse genes have indicated that choanoflagellates are the closest living relatives of animals. And then I'm going to tell you very excitingly that we've sequenced the genomes of diverse choanoflagellates. And when we compare the composition of choanoflagellate genomes to those of animals, it's very clear that they share a very close relationship to animals. Let me tell you about these organisms because you may never have heard about them before. Choanoflagellates are single-celled microbial eukaryotes. They're about the size of a yeast cell, and they have some diagnostic features that tell you that you're looking at a choanoflagellate. They have a spherical or ovoid cell body. At the top of the cell, which we call the apical surface of the cell, they have, as you can see in red here, something that's called a collar. And this is actually the source of the name choanoflagellate. The phrase choano refers to the collar, and the choanoflagellates also have a long flagellum. And you can reasonably think of these cells as resembling sperm cells with the addition of this collar. Now, choanoflagellates are actually quite diverse. They can come in many different shapes and forms. So almost all choanoflagellates have a single-celled phase to their life history, as you can see here. And as I said, all choanoflagellates have a flagellum and collar, but some of them can form beautiful colonial structures, such as you can see here. This species can actually fluctuate between colonial and single-celled. And some of them form very ornate extracellular structures, such as this beautiful organism, which can actually biomineralize silica to form a rigid structure that protects the cell and mediates its interactions with other organisms in the open ocean. Why do choanoflagellates have this combination of the flagellum and the collar? What does that do for the choanoflagellate? Well, let me show you. What you're going to see, this is a movie, and the flagellum is undulating back and forth. And what this does is it actually creates fluid flow indicated by the arrows that pulls water along the surface of the collar. And the flagellum pushes water out behind the cell. And so this has two consequences. If the choanoflagellate cell is not attached to anything, the movement of the flagellum allows it to swim along through the water column. But that fluid flow also has a second important function, and that is that it allows the choanoflagellate to pull bacteria up against the surface of the collar. And so you can see in this picture right here, a bacterial cell that's been trapped up against the side of the collar. And so choanoflagellates actually have an important and intimate interaction with with bacteria that is essential for their viability. Now, choanoflagellates were actually, although they are not widely known, choanoflagellates were actually first discovered a long time ago, in the mid to late 1800s. And people like Ernst Haeckel and William Saville Kent were obsessed with choanoflagellates. Saville Kent actually wrote a large monograph called the Manual of Infusoria, and there are many, many plates dedicated to the choanoflagellates, showing their incredible diversity. And one of the things that excited Saville Kent about choanoflagellates was that, to his eye, they were completely indistinguishable from another group of cells that he saw in the natural world, and that was in sponges. So he noticed this similarity between the morphology of choanoflagellates and the morphology of sponges, and from that, he made the argument that choanoflagellates and sponges might be closely related. And you can see that similarity, I think, even more clearly in this electron micrograph, in which you can see, again, a choanoflagellate cell with its cell body, its collar, and its flagellum. And here you can see in SEM a group of choanocytes, that's the name for the collar cells and sponges, arranged in a circle, and they're doing the same thing. They're actually creating fluid flow to capture bacteria. And I think the power or the organization of these choanocytes into this choanocyte chamber is actually a very nice demonstration of what happens when an organism becomes multicellular. And so an example of this I'm going to show you in this movie is that the coordinated action of collar cells in sponges allows for tremendous fluid flow. And so what you're going to see in this movie taken by PBS is that a diver comes in and releases a cloud of fluorescent water just near a sponge. And now watch what the sponge can do with this just through the movement and activity of choanocytes. So the diver comes in, this fluorescent dye is released near the sponge, and now as the camera pans back, you see that the sponge, which we think of as a very simple organism, is creating coordinated fluid flow. And sponges, through this action, are able to capture enormous amounts of bacteria out of the water column. So choanoflagellates and sponges are using an indistinguishable cell type to capture bacteria out of the water column. And it turns out that cells that resemble choanocytes and choanoflagellates are actually also found in other groups of organisms, including in the form of epithelian sperm. When we map the distribution of these types of cells, the collar cells, onto a phylogenetic tree, we can infer that because collar cells are widespread within animals, and they're also found in all choanoflagellates, then we can reasonably make an inference that choanocytes and collar cells were also present in their last common ancestor. And we can also compare other features of the biology of choanoflagellates and animals within the context of a phylogenetic tree, and that brings us to a very exciting point, which is that we can start to make specific inferences about the cell biology and life history of the first animals. So in this schematic, what I'm showing you is what we now infer to have been the case for the biology of the first animals. We think that it had a simple epithelium, this planar sheet of cells. We think those cells were adhering tightly to each other. We think that some of those cells, at least, were capable of differentiating into collar cells, and importantly, that those cells were actually eating bacteria. So the first animals were bacterivorous. We think that the first animal also was capable of undergoing apoptosis, or programmed cell death, and that there were different cell types in the first animal, indicative of cell differentiation within the soma. Moreover, it's become clear by looking at the distribution of different modes of sexual reproduction, sperm and egg in animals, it's become clear that the first animal from which all living animals evolved was capable of undergoing gametogenesis, and that it produced differentiated eggs and sperm, and that these merged in a process of fertilization to produce a zygote, and then that zygote underwent multiple rounds of cell division and cell differentiation to produce this adult form that I just told you about. So I think this is an exciting time in which we're starting to see the power of comparative biology, and we can compare the cell biology of choanoflagellates to animals and start to really make specific inferences about the biology of their last common ancestor. Moreover, with the advent of genomic approaches, we can start to learn something about the genome of this organism. Now, choanoflagellates have really been relatively poorly studied by molecular biologists. There was this flurry in the mid-1800s in which people were spending a lot of time looking at and thinking about choanoflagellates, and then they were relatively forgotten within the world of molecular biology and during the molecular biology revolution. And so one of the first things I did when I started studying choanoflagellates was to collaborate with the Joint Genome Institute and the Broad Institute to sequence the genomes of two different choanoflagellates, Montasiga brevicollis, which so far we have only seen in unicellular form, and S. rosetta, which can be single-celled or colonial. These genomes have a modest number of genes, between 9,000 to 12,000 genes in their genomes, and we can compare the composition of those genomes with animal genomes to make inferences about the genome of their last common ancestor. In addition, we've recently started sequencing the transcribed and translated genes in the genomes of 20 other additional choanoflagellates that are in culture. And I just want to make the point that there's a lot of diversity in choanoflagellates, and by surveying the genomes of many, many different choanoflagellates, we're starting to get an increasingly complete and complex picture of what the genomic landscape of animal origins might have been. Now, I'm not going to tell you about all of the different genes that are found in that ancestral genome, but I do want to summarize some of the exciting findings. When we analyzed these genomes, we particularly focused on genes whose functions are required for animal multicellularity and animal development. And in particular, we focused on genes that are required for cells to adhere to each other, genes that are involved in cell signaling, that is, allowing cells to talk to each other and coordinate their functions, genes that are required for gene regulation, which allows one cell to differentiate its function from the other, and genes that are involved in interactions with what's called the extracellular matrix, the ECM. And these are the genes and proteins whose functions allow cells to create this matrix, this structure that provides a landing spot and scaffold for cell-cell interactions. So we can think about these as being essential functions for animal multicellularity. Many of the genes that are required for these processes in animals had not previously been found in a non-animal before. And now we can ask, if we look at choanoflagellates, what does that tell us about the ancestry of these genes? Are they really animal specific, or might some of these genes have evolved earlier to serve other functions? Now remember, we have to do this within a phylogenetic framework, and so we're going to ask two different questions. If we are focused on these classes of genes, what fraction of them seem to be restricted to animals, and what fraction of them are also in choanoflagellates, and therefore we infer present in their last common ancestor with animals? Some of these genes might have evolved much earlier in the colonial and unicellular progenitors of animals. So when we do these types of comparisons, and when we did them, it was really quite exciting. I think it helped to motivate a lot of the future study for choanoflagellates, and that's because choanoflagellates turned out to express many different components of many different genes that were required for the functions that I was just discussing. So we can find genes that are required for cell signaling in animals, including things like, it's a bit of a chicken soup, but the GPCRs, these are G protein coupled receptors, the receptor tyrosine kinases, proto-oncogenes like SRC and CSK. We can also find genes whose functions are both necessary and sufficient for allowing cells to stick together. These include the cadherins and C-type lectins. We can find representatives of various transcription factors that are involved in gene regulation, MYC, P53, and forkhead. And we even find genes that are involved in forming and coordinating the interactions of animal cells with an extracellular matrix. But remember, we're finding representatives of these genes in non-animals, the choanoflagellates. And so I think an exciting future area of research is to try to figure out how these genes function in choanoflagellates and try to make inferences about how they might have functioned in our long ancient progenitors. Now, it was very exciting to find all these animal genes in choanoflagellates, but I think we all need to agree that choanoflagellates are not animals. So what makes animals different? And what is exciting is that these genomic interactions, these genomic comparisons, allow us to learn about what types of genes and genomic innovations might have actually contributed to animal origins. And so when we look at the gene complement of animals and compare it to choanoflagellates, we find that there are some genes that thus far have never been found in a non-animal. And so these are representatives from each of these different groups of processes as well, and they include important genes involved in developmental signaling, one special class of cadherins, the classical cadherins, that are essential for allowing epithelial cells to interact. Important and famous developmental patterning genes like the Hox genes so far have never been found in a non-animal, and very specialized forms of extracellular matrix components, including the type 4 collagens. So this having genome sequences from living organisms has now allowed us to reconstruct in increasing detail the genomic landscape of animal origins. So what I want to say then, and what I've tried to say in part one, is that by studying these previously enigmatic organisms that had been poorly studied, we're starting to grow and develop a new model for animal origins. And we can study these organisms now in a modern context to start to learn about animal origins and details. So what I've told you in this first section is that choanoflagellates, the study of choanoflagellates, has illuminated the cell biology and genome of the progenitors of animals and told us that those first animals probably ate bacteria and had collar cells. And the second important thing that we've learned by studying choanoflagellates is that a remarkable number of genes required for multicellularity in animals actually evolved before the origin of multicellularity. And an exciting future area of research will be to figure out what those genes were doing before they were required for mediating cell-cell interactions. So that is the completion of part 1. And in part 2, I will tell you about a transition to multicellularity that didn't happen hundreds of millions of years ago, but actually happens every day in one particular choanoflagellate. And I'm going to tell you about how that's regulated. Finally, this work wouldn't have been possible without the help of my past and current lab members. And I'm also very grateful to all the collaborators that have made all of this work possible. Finally, I'm very grateful for the generous support that's come from the National Institutes of Health, the Gordon and Betty Moore Foundation, the Canadian Institute for Advanced Research, and most recently, the Howard Hughes Medical Institute. Thank you very much.